Thanks a lot for showing up this morning. I'm going to get things started and, spend the first 18, 20 minutes. I'll try to keep it more towards 18 to go over some of the basics and share some of the data that we have had people publish with our instrument technology workflow that we're going to talk about. I'm Deb. Last name is way too long. As I always say, my parents didn't quite like me, so they gave me this. Without further ado, essentially, this whole technology of adaptive focused acoustics, it's basically an acoustic energy that goes and hits the sample at a specific frequency. And it's a short wavelength, long range frequency that it goes and targets to the samples. And the sample that is sitting in a buffer, which we all provide because it's in the form of tissues, it's in the form of cell lysates. That's in a 96 well plate or a single vial or an 8-strip format, where you create a lot of these cavitation bubbles because of the energy hitting the sample, which in turn coalesces, implodes, releasing an energy that causes samples to disrupt, shear. So that's essentially the first step when you start with tissues, cells. That's where you start the game, because you want to extract as much protein as possible. Now, you would say that, okay, that's all good, but where does it actually make a difference? Because all you're telling me is I'm doing a sonication. Well, I have already indicated a specific frequency, which makes it very, very effective. And you can see it actually on the slide over here, is that typically you would have a lot of pressure coming to the sample and heat generated. Now, for everybody in the room here, it's quite obvious you all are associated with proteomics or protein-related analysis, and in that case, heat is not the biggest friend you can ask for. And as you can see on the bottom of the screen, this is where this technology works, which is basically controlling the temperature. So you get to keep it cold or warm or somewhere in between that, no matter which direction, whatever samples you're working with. So for more than 2 decades, this has been the go-to technology for most next-gen sequencing lab for doing DNA shearing. But, 5, 6 years ago, some of our users thought that's only half the story. It's in systems biology, we are only talking about genomics, chromatin shearing, working with DNA, working with RNA. That's only half the story. And the fun part is where it gets a lot more complex as we move to the other part of it, which is working with proteins. So in doing so, the next question is that why bother? The reason why it is very important is because in the world of proteomics, there's actually a lot more happening now than what has happened maybe 10 years, 8 years ago, where actually things are stepping beyond just research. People are moving towards implementation into biomarker-related studies, targeted proteomics, even targeted protein quantitation. You look into the biopharma field, especially, it's fair to say 40% of drugs or therapeutics that are approved by FDA on a year-to-year basis are actually large molecule-based drugs. So they're all, you think of Humira, you think of Kadcyla, these are all some monoclonal antibody-based therapeutics. So that is very targeted, but then across the board, when you look at different kinds of diseases, there are a lot of these protein-based therapeutics that are actually becoming very, very important in terms of biomarker-based analysis. And actually, I would not go too much into this because I don't want to steal Saeed's thunder, and he's going to go and focus on specific application areas that he and his lab has been invested in, focused on. But what we have developed is basically a one-plate workflow. So essentially, I mean, during my PhD days, a part of my PhD towards the end actually was spent in Louisiana, the last 7 months or so, and there was a catfish place over there that said, live at one end, fried at the other, right? So, and when I see this, it kind of tells me something similar, no catfishes here, but it's hard samples, difficult samples at one end, and clean, pure peptides at the other. So you drop your tissues, you drop your, put your cell lysates, you work with all of those things and into the 96 well plate, into the 8-strip tubes, and you perform your cell lysis, tissue disruption, all in there, and then move on to the next stage where you do the immunoprecipitation, you do your binding, and then you do your digestion on bead or off bead, and you work with your peptides, eventually getting it into your C18 column or straight into your LC. That's what we are offering to do in a form of a one-plate analysis, which I think is actually very cool because typically taking sample in and out can result in sample loss, but a lot more than that, how much do you stand to lose in terms of your peptide cleavage, your digestion mechanisms, so on and so forth. So you don't necessarily have to take my word and all these positioning slides that I'm showing, because there are people who have actually presented quite a bit and published their data quite a bit. In this particular example, there's this lab that has presented the data, and it's published here. You can get the reference from the bottom of the screen, where they're looking at more than 1,800 proteoforms. So they're actually doing intact. Now, here comes the question, because you might say that, hold on a second, you said I could disrupt tissues, I could lyse the cells, I could basically break the samples down to, smithereens. So they're working with proteoforms. Is it controlled? And the answer is yes. So essentially, you do not necessarily change the frequency, because that's our secret sauce, but you get to control the total amount of power that you are using to disrupt. And at a lower power, you don't necessarily break everything down into the smallest fragments, which is why we once again say, and you will see this data again and again, during Saeed's presentation, I'm assuming, that we also see in presence of this particular energy hitting the sample when you're doing digestion, it expedites the whole digestion process efficiency without adding any additional miscleavages or any additional post-translational modifications. Because you get to control the power, how long you switch this power on, whether you apply it more like a pulse, or for the LC aficionados, if the power is more like an isocratic. So all those things are in your control. So that's what this group has shown, and also from a bottom-up perspective, the previous slide actually talked about it, how we can enable taking the samples from the start and all the way going to peptides. So here is another example, which is basically where this team is focusing on working with both proteomes and metabolomes, not necessarily out of the same sample, but happening it, working on it together. So basically they extract the sample, they perform the proteomic workflow. For metabolomics, they use a different workflow, of course. And where I think it makes a big difference is where metabolomics can come into play, because this thing is very fast. We are talking about few seconds to maybe a minute of exposure to this energy that can actually cause high amount of extraction of proteins, metabolites, DNA, RNA from your samples, and then further processing of those. So here is another example where actually they looked at impact of diabetes on cardiomyopathy. And so this group is based out of Manchester, United Kingdom, where they looked at different kinds of tissues, also organoids, small cardiac muscle pieces. So they started with that and went all the way to monitor specific proteins that they used as markers to see their expression, how much they were upregulated based on the status or the health situation that the patient was in or the patients were in. And the whole platform that they were using were actually this sample prep platform that we offer. And I'm not going to talk about the product names because we are all here until Wednesday to tell you about the product names, show you the products if you come to our booth. So I do not want to take your mind space by highlighting just the product names, but if there is one thing that I would really, really like for all of you to take from this part of the presentation is take your complex samples and get reproducible, reliable, clean peptides at the other end in one sample prep workflow. So here is another example where we show how we actually significantly reduce the total TMT process. So starting with whatever number of 50 steps plus 48 hours or 48 hours of time that people take to go through the entire process, it comes down to somewhere around 20 steps and 5 or 6 hours of workflow that you do by expediting the entire process without compromising the quality of data. So in this regard, some of these examples where people actually worked with cell sorters and they divided the samples into 384 cells and they did the whole workflow using the cartoon that you're seeing at the bottom of the screen is actually one of our instruments, and they did the entire process once again, the lysis, DNA shearing, which is what you have to do anyways to extract your proteins, and the whole process was done leading up to when you inject your sample into LC-MS. So this is completely agnostic of whatever LC or MS systems that you are using. Of course, in all likelihood, most people over here you all are working with high resolution accurate masses, but it also works with triple quadruples, so that doesn't really make any difference. And here is one of the major thing that in my opinion is a very cool aspect. So on a show of hands, anybody over here work with FFPE? Okay, one hand I see going up, that's fantastic. So typically, and I'm now speaking to you directly then, since you raised your hand, you probably use xylene or some sort of a mineral oil or some sort of mechanism to remove the paraffin. Now, this particular capability or technology actually deparaffinizes, emulsifies the paraffin on its own without addition of any additional organics. So you put your FFP scroll and it emulsifies the paraffin, exposing the tissue. Then you use the same technology, same plate to disrupt the tissue, and then you eventually get your proteins and go through the whole process. So there's been a lot of work, and on the DNA RNA side, we have a complete automatable workflow that starts with FFPE scrolls and gets the DNA, pure DNA and pure RNA out in about 5 hours, bringing the whole workflow down from close to 16, 17 hours down to about 5 hours of total workflow time. So we have actually got all these things in publication, but before even I get into that space, I would like to highlight that this was actually, I would say 4 years ago, Matthias Mann came up with the first publication where he started with FFPE as well as fresh frozen tissues, and he went through the whole workflow, which was published over here. And so much so, he kind of did the whole thing on an automated workflow, using an Agilent Asimov Bravo. So the entire perspective from tissue deparaffinization, tissue disruption / lysis, and the whole workflow can be done, and resulting in him reporting out all the additional peptide IDs that he found as a process, also the higher yield of proteins that he got. Now, one question that might come to your mind is that, oh, is my yield going to be higher every single time? And it depends on the workflow. It depends on the proteins you are trying to work with. And of course, we have got a very talented pool of scientists in our team who actually are very diligent and knowledgeable to help you with that in optimizing your workflow. And Samir is sitting right over there. I actually don't know why I'm talking with him sitting over there. But in general, that's where we come in, because if you would be working with transmembrane proteins or lipophilic proteins or hydrophilic proteins. So either way, there are certain nuanced changes that we can recommend to ensure highest amount of extraction of your proteins, whether it's targeted or global proteomics, from your samples of choice. However, there's one thing that you are bound to have, which is basically the reliability and reproducibility of your assay every single time. So I'm almost towards the end because I really wanted to give Saeed most of the time to go through his presentation and highlight all the cool data that he and his lab members have achieved over their whole workflow where our instruments play a part. So I showed FFPE, I talked about tissues, and here is one slide that kind of captures different sources of tissues, different kinds of variants, starting with essentially the SMA kind of tissue, which is basically, you get, you use those tissues for your phenotype assays, to brainstem tissues. So basically you work with brain cells or brain cubes or brain tissues obtained from brain, to all the way to skin tissues. Now, so what I'm highlighting over here is basically the complexity of the samples can vary significantly. You can have something very soft to something that has got a lot of adipose, a lot of lipophilic characteristics, to something as hard as skin tissues. No matter what you are using, you will be able to extract the maximum amount of proteins and do the entire protein binding and digestion workflow until you get to your LCMS. So we have actually in our bag a lot of workflows that are already optimized with STRAP, with SP3, non-SP3. A lot of these things are actually already well optimized, and we have those workflows, methods already well established on our, available on our website. Plus, as I mentioned, we are always willing to discuss with you and incorporate those nuanced changes that would give you your biggest bang for the buck, so to speak. And here is the total lineup of the instruments. For most practical purpose, I prefer focusing on the last two guys on this screen, which can take on the 96 well plate, or also it can go to potentially holding a 384 plate. Also, it can take 8 strip tubes, and both of them have that ability to work with the holistic, the comprehensive protein workflow. We provide all the consumables. We do provide reagents if you would like us to, but we also are more than happy to work with whatever workflow reagent kit that you are using today to ensure that you actually get the highest amount of efficiency that you are looking for, aspiring for. So as a result, basically I mentioned, you are looking at robustness, reliability, reproducibility, because if you don't have that, if your person CV is high, it's unlikely you're going to get much reliable information out of that. Addressing matrix complexity, having that confidence in working with your samples, and applicability across the entire system biology, from DNA shearing, RNA shearing, proteomics, metabolomics, transcriptomics. This is actually being used across the board for a host of different kinds of applications. More than happy to share with you details of those. And so we are actually here to think about, whenever you go out and you think of Covaris, when you see Covaris, you will see, oh, it's the DNA shearing company. Keep in mind that we are actually a sample prep company. We are in the business of providing sample prep not necessarily a technology that does one particular thing very well. So once again, complex samples to clean, pure, high yield of peptides. That's what we actually are here for. We are actually at the booth, which is at the corner in the room. If you want to discuss your specific needs, please come by. We will be there. We are more than happy to talk to you. And, of course, we are more than happy to build the pool of people so that next US HUPO, I am not the one talking, one of you would be talking, so that, we are all looking at new sets of data. Having said that, you are not here to look at me and to listen to me for the whole hour. Not a very pleasant sight. So, but there's someone who actually is going to provide some very cool data and go through the examples that he has developed over the years in his laboratory. So without further ado, Said, thank you so much for your time. We'll have some time to have questions towards the end. Thanks.